All of the cells in our bodies were once pluripotent stem cells. Pluripotent stem cells have an amazing potential to give rise to all of the body's tissues and can follow many different pathways to become specialized cell types such as neurons, muscle cells, red blood cells, and macrophages. Stem cells are not only important for development, but could also be extremely useful for therapeutic purposes, and researchers hope to use them to replace diseased tissues. Some stem cell therapies are already being used in the clinic. Controlling stem cell behavior in vitro or in a lab dish is a vital step toward developing therapeutic applications. Once a desired cell type has been obtained from stem cells, doctors could transplant the new cells into the patient. Using a patient's own cells avoids many of the problems of organ transplantation, like rejection by the immune system and the need for a donor. Pluripotent stem cells are therapeutically useful because they can differentiate into any other type of cell in the body. However, they are not easily obtained from adults. In 2006, Shinya Yamanaka, a scientist at Kyoto University in Japan, came up with a method for turning committed adult cells from mice back into pluripotent stem cells. These reset cells were called induced pluripotent stem cells, or iPS cells. These iPS cells should be functionally identical to embryonic stem cells, cells found early in the developing embryo that have the potential to differentiate into any cell type. Since iPS cells can differentiate into any cell type as well, it is possible to generate a complete adult mouse from a single iPS cell. iPS cells are a more ethically acceptable alternative to embryonic stem cells because they do not require the use of an embryo and can be obtained through non-invasive procedures in adults. iPS cells also allow for the creation of a personal pluripotent stem cell line for each patient.
There are a number of steps on the journey back to pluripotency. First, we begin with a mature adult fibroblast cell, which is easy to extract from a sample of the patient's skin. We need to introduce 4 transcription factors, proteins that regulate the expression of other genes needed for pluripotency, into the cell and drive their expression. One way to do this is with a type of virus called a lentivirus, which can transfer the pluripotency genes into the genome of the adult cell. The lentivirus with the pluripotency factors also contains a sequence that allows us to turn these genes on or off at will when an antibiotic called doxycycline is added. When we add the lentivirus to the cultured fibroblasts and then add the antibiotic doxycycline, the 4 pluripotency factors are expressed within the cell, and the cell returns to a pluripotent state, ready to become any cell type in the body. Once fibroblasts have been reprogrammed to iPS cells, they behave like embryonic stem cells, express genes characteristic of pluripotency, and can be differentiated into any cell type in the body. The next leg of the journey is differentiating the iPS cells to a myofiber. To transform a pluripotent iPS cell into a differentiated cell type of our choice, we need to express a gene that drives the iPS cell down a certain developmental lineage. We can do this in much the same way we generated the iPS cells in the first place. We introduce a lentiviral vector containing a gene called MyoD1, which causes differentiation into skeletal muscle. We then add doxycycline, driving expression of the MyoD1 gene. When allowed to mature, these cells will fuse together to form muscle fibers, which twitch in the dish just like real muscle. The ultimate goal of regenerative medicine is to produce healthy cells that can replace diseased tissues in the human body. In the near future, doctors hope to take a sample of a patient's adult cells, induce pluripotency, guide the differentiation of these cells to the desired cell type, and use these cells to treat the patient's disease. Stem cell research is progressing quickly, and the future looks bright for regenerative medicine.